Blotting techniques in molecular biology. Blotting refers to the transfer of macromolecules, DNA, RNA, or protein, from a gel to a membrane, followed by detection using labeled probes or antibodies. These techniques are essential for identifying specific biomolecules among complex mixtures. Types of blotting techniques. Technique targets probe type used for southern blot DNA or RNA probe detecting specific DNA sequences. Northern blot DNA or RNA probe studying gene expression, mRNA. Western blot protein antibody detecting specific proteins. Eastern blot post-translational modification antibody or chemical probes identifying modified proteins. Southwestern blot DNA binding proteins labeled DNA probes studying DNA-protein interactions. 1. Southern blotting, DNA detection, developed by Edwin Southern, 1975. Steps: DNA extraction, restriction, enzyme digestion of DNA, agarose gel, electrophoresis to separate fragments, denaturation of DNA in the gel to make it single-stranded, transfer to membrane, usually nylon or nitrocellulose, hybridization with labeled DNA, RNA probe detection, e.g., chemiluminescence or autoradiography. Applications: detecting mutations or deletions, DNA fingerprinting, genetic mapping. 2. Northern blotting, RNA detection, purpose: to detect and analyze mRNA and study gene expression. Steps: RNA isolation, denaturing gel, electrophoresis, to prevent secondary structures, transfer to membrane, hybridization with labeled probe, complementary to RNA, detection. Applications: measuring gene expression levels, detecting RNA viruses, comparing mRNA between tissues or conditions. 3. Western blotting, protein detection, also called immunoblotting. Steps: protein extraction, SDS page to separate proteins by molecular weight, transfer to membrane, PVDF or nitrocellulose. Blocking, with BSA or milk to prevent non-specific binding, primary antibody incubation, specific to target protein, secondary antibody incubation, labeled with enzyme or fluorophore, detection, chemiluminescence, fluorescence, or colorimetric. Applications: confirming protein expression, diagnosing diseases, e.g., HIV, identifying post-translational modifications. 4. Eastern blotting, modified proteins, purpose: used to detect post-translational modifications like lipidation, glycosylation, or phosphorylation. Steps: similar to Western blotting, but involve specific chemical or lectin probes for detecting modifications. 5. Southwestern blotting, DNA-protein interaction, purpose: used to detect DNA-binding proteins like transcription factors. Steps: proteins are separated via native page, transferred to membrane, labeled DNA probe is added, interaction is visualized. Applications: studying gene regulation, identifying unknown DNA-binding proteins. General materials used in blotting: membranes, nitrocellulose or PVDF probes, radioactive, fluorescent, or enzyme-linked detection methods, chemiluminescence, autoradiography, or colorimetric substrates. Applications of blotting techniques: diagnosis of infectious diseases, HIV, hepatitis, oncology, detecting oncogenes, tumor suppressor proteins, gene expression studies. Research in cell signaling, apoptosis, and protein-protein interactions. Forensic analysis. Mnemonic to remember: SNW, Southern, Northern, Western equals DNA, RNA, protein.